That's okay. Okay. So I'm going to read questions that are coming from our colleagues who are listening online. This is from Anna Mastroianni. So, to Dr. Liang, can you clarify the confidentiality protections provided to the applicants by the FDA when an IND is submitted? That's one. And second, what regulatory authority does FDA have to require long-term follow-up of clinical studies?
Can you repeat the first question?
Sure. Can you clarify the confidentiality protections provided to the applicants by the FDA when an IND is submitted?
Yeah, so we're not allowed to discuss confidential information, so we can only discuss the published data.
Okay, but the identity of those who are applying for INDs, I'm just kind of trying to read between the lines of that question. So, what is considered confidential and what is considered public? Maybe ask it that way.
Okay, so if there's no any published information on the website, and that is confidential, so we cannot identify a specific applicant who apply any clinical investigation. So as long as there's no any publication or announcement from the website or from any specific sponsor in their website, we cannot discuss the identity of the applicants.
Okay, and then the second question was the second question about the FDA.
The long-term follow-up.
Yeah.
Okay, so as I mentioned in my slides in my presentation, so the purpose for the preclinical data is to support the rationale for the clinical investigation. Safety is always primary. So the aim for the long-term follow-up is to adequately catch any chronic toxicity or adverse events. So this is required by the regulation, and so for as long as if the study duration should be long-term in order to catch any chronic toxicity, then we need long-term follow-up.
Mm-hmm. Yeah, let me look at it again.
She's got another one coming in too. Regulatory authority.
Yeah, so the question is really about long-term follow-up and what can the FDA require as a matter of jurisdictional authority, if anything.
I'm not sure. Okay.
Okay.
He may be referring to phase 4 studies.
You want to? Well, it's coming from Anna. So the question is sort of, during phase 4 or wherever you might see an appropriate place. And maybe Celia, do you want to jump in here? That would be fine too.
There's a microphone.
There's a microphone right behind, right here, right behind you.
Oh.
Yeah, there you go.
Okay, so just to clarify, I think the question actually relates to requiring long-term follow-up in the clinical study.
Correct.
And not in the preclinical study.
Correct.
And so the question really is, well, we can require things to be added to a clinical study, but actually requiring someone to be able to carry it out is hard to do. Because if they, just think about it, if the sponsor wants to close the study or go out of business, or the patients, as the previous speaker says, doesn't want to be followed, that is just not something that we are able to do. So I think that is the answer to that.
Okay.
Thank you.
So maybe you can stay here for this is a kind of follow-on to the IND confidentiality discussion. So.
Also, I just want to clarify, I think it was clear in Wei's answer, but just in case it wasn't, we can't even acknowledge the existence of an IND.
Okay.
So it's not just whether or not who the sponsor is, but does it even exist? We do not acknowledge that.
Okay, so that may answer this second question. This question that's a follow-up about that, which is, what if any opportunity for public discussion is there when an IND is submitted, and in particular, I mean, just none, I guess, is the answer.
That is correct.
Yeah.
There's none.
Okay.
So we have another one for John.
So yeah, so now I'm gonna one more from Anna, and then I've got Keith on my list. I can read that. That's actually easier for me. Thank you. This is for John Gearhart. So this is in sort of a question around your statement about the 17-year gap between the beginning of the basic science and maybe moving into applications and therapy. How would you allocate that time frame in terms of typical time for scientific exploration versus the regulatory environment versus the political and social environment, in particular in the stem cell research area? And so this is just your sort of perception, and in your opinion, is there a mechanism or regulatory approach or review process that could have expedited the time frame to make the field move forward more quickly?
I think if we had an hour, we could probably cover all aspects of that. Clearly, in the beginning of it, there was a great deal of uncertainty as whether work could even be continued. At what institutions, under what facilities could you use? Funding, let's be honest about this, for most of us, if it wasn't for philanthropy, this field would never have really exploded. So there's an issue of, obviously, in delay, as many institutions sort of took a breath and trying to figure out what the federal regulations were, if any, that would be around this work, to ramp up getting the necessary funds in place. So there was a good deal, I think, of delay, or at least not getting out of the box on this. And then, like any other biologic, entity, when you're trying to, particularly with pluripotent cells, when you're trying to derive cell endpoints, I mean, whether they're cardiomyocytes or anything else, we're still debating whether we have normal cells in culture out of this. And this is a big deal when we're now beginning to see people moving towards trying to get clinical use of this. These arguments, how can you prove that this is a normal cell that will, behave when you want to, and then to go through this process with GMP and whatnot. I mean, this is terribly expensive to move in that direction. So I think there's some biology in there. Obviously, there's issues of the regulatory things. I think from the regulatory side, and I think something similar to MRT, is I think we have to have a change in pathways here, where we begin to look at the safety issue and the concern for long-term safety. When you put a cell into somebody, how can you assess from animal studies or anything how safe that's going to be in a human? And to me, this is very difficult, and I think this is a bit of a stumbling block. And even in monkey work that we're doing, and we have cells in there for a year, or is this sufficient time to say they're going to be safe? So I think it brings, it's a combination of,
some of each.
All of those things, yeah. So, yeah.
I've got Keith and Jeff Botkin and Vamsi and Alta. Keith.
So a quick question. I have to admit my head is sort of still spinning because of a number of comments made about there being clinics set up to already do what we are discussing, just not in the continental United States. And the image of your concern about the world anti-doping, the way in which both stem cells and these techniques are likely to be employed offshore. And then I'm kind of also find a compelling yet confused image of this moving along three lanes of a highway, moving in different directions. So a swirl of kinds of real questions and metaphors. And so, but one of the things that connects a lot of your comments is the fact that much of the investigations for this line of this inquiry is happening elsewhere. And so I guess I have a question for the three of you, which is, how should this committee think about and learn from non-U.S. developments, and also within U.S. disparities, the ones that you highlighted, the fact that, you know, we have different policies from Oregon to Massachusetts to California, New York, restricting egg donation for research. How should the committee think about, I mean, you can argue that we should learn from the UK, you could argue we should not learn from the UK, you could argue we should learn from what's going on elsewhere, the fact that patients are going elsewhere for these services, or you should say, well, that's no reason why we should embrace these. So I'm just interested in your observations about how what are the lessons that we should take from these developments for the work of this committee?
I think there are a lot. You know, you heard reports that the UK has, you know, these you have them in your materials, the 3 reports from the science, the issues of ethics. That if you read those very carefully, and maybe the people around the walls haven't read them, there is a lot of material. It's current, it's accurate. I think the perspectives that they have on this have proved to be right from the beginning. Now, whether or not the U.S. would set up something like an HFEA, I know Alta has some very good wisdom on this. It will never happen. But there's a lot in there that I think we should be able to look at and could help you from that standpoint. I know of no other country that has this kind of, you know, complete history and have been there before us to a certain degree. I think at looking at some of the regulatory things that have changed in Japan and Korea for fast tracking some of the stem cell kinds of things. Now, I would say maybe they're driven by more economic matters than a Nobel Prize, I don't know, to try to recapture, you know, some of this. That may be worth taking a look at. I think I want to clarify a point. I know of no offshore clinic that is engaged in any type of human cloning or somatic cell nuclear transfer or MRT. I mean, I'm not meaning to imply that I know of a lot of other clinics that are doing other things, okay, in it. The final comment I would make is that I wish the committee would have a uniform policy. No matter what states say, I think there should be some kind of a federal policy, and whether a state chooses to do it or not, that's their issue. This helped us to some degree in the stem cell issues. You know, the state of Pennsylvania will not permit you to establish cell lines, but you can work on already established cell lines within the state. And that was a great, and the fact that there was a federal policy on this really, I think, calmed down some of the legislators who were thinking of, you know, trying to stop it all and saying, well, the federal government has looked into this. They have a relatively good regulatory thing, and they permit it to go. And so I think you got to be a little. I know you may be sensitive to other states' things, but I think a more uniform policy would be in place. Should be through the country.
Insoo, do you want to?
Yeah, sure.
Right away.
Yeah, so I ran out of time, and I couldn't really quite finish my 3-lane metaphor, so I'm not surprised people are kind of still spinning from that. So what I wanted to do with that metaphor is the following. I mean, you have to realize that in the discussions we've been having and that you'll see outside of this room, clearly there are 3 attitudes about how we should proceed with scientific technologies. And I just want you to be aware that it seems to me that the FDA is kind of already built in a way to be on the left lane. And a lot of the concerns that I've been hearing from many people are the right lane concerns. Technology selection, are there alternatives? We really need to do this, manipulation stuff. What about adoption? I mean, those are exit. Yeah, the nearest opportunity, because driving is dangerous. I mean, that's the. And I think that in the U.S. there are different populations of people in each lane. So what am I doing with this metaphor? So in the UK, arguably, there is some sort of infrastructure there to deal with these kinds of questions, right? So we have a national body that reviews embryo research and advances in ART. There's the whole consultation process. I mean, arguably, there are some roads and some traffic cops and some signs there. I think in the U.S., we have just a big old open field. And what I would suggest is there needs to be more like regulatory capacity building before we just start launching straight ahead. Should you use the UK as a model or a template for the road work in the US, the regulatory infrastructure? I'm not sure. I mean, there they have right-hand drive, we have left-hand drive. I mean, maybe there are some important differences between the two, okay? The populations may be different. The UK is, as far as I understand it, not quite as religiously sort of driven as the US in terms of policy. That might just be my misperception. But anyway, that's what I wanted to do with the metaphor. There are choices available. Now, in terms of your committee, I think you need to think about, because when I look at the list of questions you're supposed to be pursuing for FDA, I mean, they're kind of a blend of like the different lanes. So I just thought, this might be kind of a way for you to kind of think through at what levels you're going to address these concerns, and not wedge yourself, I'm not saying that you're doing this, but not wedge yourself too much to like presenting a set of recommendations that already presuppose we're only talking about left lane stuff. So that's what I was hoping to do with the metaphor.
Thanks. Jeff Bodkin.
So this question probably both for Wei and Insoo. How does FDA decide when it has sufficient preclinical information about safety and efficacy to allow first-in-human trials? So we've heard that, we have some animal data out there. Some folks have said we don't have enough. Maybe we should be doing cows. How do you decide, based on the number of species, perhaps the outcomes of those preclinical trials, that it's okay to then move to humans? And then maybe the question for Insoo is, maybe when should we think about making that move? And does the fact that countries around the world are beginning to use this in humans, how would those human trials elsewhere influence the FDA's determination about first-in-human trials in the U.S.?
So I think regarding the adequacy of the preclinical data, including the in vitro and in vivo studies, we have discussed these issues in our last year's advisory committee, and we do take those advice into our consideration if we receive any application. So how do we decide how much is sufficient? I think that really depends on the specific product and technology, or the proposed clinical indication and its risk-benefit profile. So for a specific product, if this the population, the proposed indication is let's say it's a life-threatening disease, then that will the risk-benefit ratio will be different from another indication that is not life-threatening condition. And so for previous human experience in other country, this is not only this may not be only unique for these MRTs, but also we have some other experience with cellular and gene therapy products that we regulate in our office. So we do consider those previous human experience, so we can get some general picture of this overall safety and activity of a specific product. But we may or may not agree to the conclusion from other foreign agency. But that will inform our decision. We do take that into our consideration.
So yeah, to answer your question, I'm going to go back to my lanes because it depends on which lane you're in. Okay, so when do we go from animal studies to first in human? If you're in the left lane, you think, well, at some point there's some level of less uncertainty, and we've done enough studies, and we'll do trial and error. We'll go ahead in the human case and track that. So that's one approach. The other one might be, there's never a point where you can guarantee that you've freed yourself from scientific uncertainty, because that's intrinsic. Uncertainty is intrinsic to science. So you have to weigh other factors, things like, so like the risk-benefit ratio, or, are there any acceptable alternatives or not. You bring in other values in the decision-making for the crossing that line from animal study to human. In the right lane, I think, again, people are going to insist on, it's too early to even think about that, but we need to explore more of these other options, let's say PGD, or we need to be more sort of proactive in choosing which technologies we want to pursue, and that's going to be determined by the controllability of the vehicle, controllability, reversibility of bad effects. Those are the kinds of considerations. So depending on which lane you're in, you're going to have sort of like different standards for go, no-go. You're going to have like different signals, different tracking signals. So you have to complicate my metaphor even more. It's a highway with different signals, different signs per lane. And then the question is, well, which one is the best one? That's a philosophical question. It's not a scientific question. Which lane is the best lane? Yeah, is it snowy? Is this Cleveland or is it Boston or the potholes? Cleveland or potholes,
We're
right?
heading down a path here.
Don't go too fast. You're going to break a rim.
Yeah. So just a point of schedule. We need to finish this session by 11 because we have time constraints for the next speakers who are joining us via WebEx from different parts of the world. So that's just FYI. I've got Vamsi, Alta, and Jonathan, but that means we need to be concise in our questions and our answers, please.
I have I think, a quick question for Wei, and that is, we heard from John Gearhart as well as from the last few days that these are, the proposed procedures are technically very challenging, and there's probably very few people in the United States or centers that could perform this, and at least based on what's been published, there may be only 2 places in the U.S. even, based on published literature, that are capable of doing MRT. So if somebody else puts in a proposal to perform MRT based on what's been published by these 2 groups, would the FDA consider that research proposal? And how do you ensure that those individuals have the technical expertise with which to replicate what's been reported in the literature? And a close sort of second question related to that is, if a particular center receives the green light to move forward, and then the technician that had the great skill with which to do this leaves, and then somebody else comes in to do it, does the FDA somehow regulate who the operator is that's doing the procedure to ensure that they have the technical acumen?
Okay, that's a good question. So first of all, if an applicant approach us saying that they will do such MRT and based on published data, and we will not just accept published data from other investigators if this person is not directly involved in the cited publication. So a specific facility should have their own feasibility data to convince us that they can also do this technology in their own facility. And why do we regulate the operator? So I think that's like a moving target throughout the any product development program. So at any stage, we need to make sure that for this case, it's really special because this is a complicated technology. We need to make sure that any technology, any procedure can be reproduced throughout the product development program.
Alta.
All right, I'm going to try to be very quick. I just want to check my understanding of something, okay? Insoo, you made the interesting point that if the FDA were to require preclinical work with human material, that it would be preclinical work that involves making an embryo, would therefore not be eligible for federal funding, so that we'd once again have private funding. However, at the point at which you started human trials, right? This is why I was fussing around here. Dickie Wicker says you actually can work on fetuses and therefore on embryos if it is therapeutic. And we've had this interesting point about whether mitochondrial replacement versus mitochondrial modification through, let's say, gene editing has any real difference. Wouldn't it be therapeutic to the embryo to actually try to replace the mitochondria, unless we're going to go down Dr. Bredennord's, kind of black hole of identity analyses, and the replacement makes it a different identity, so it's not therapeutic to the same embryo. But am I correct that the clinical work now could be federally funded and be kind of a reversal, right? It's private for preclinical and federal for clinical, exposed to the usual order of funding?
No, I think it depends on how you interpret the second part of the Dickey-Wicker Amendment, which is it cannot be greater. Risk of injury, death, all that can't be greater than that allowed for research on fetuses in utero.
Right, which is what I just looked up. And you
Yeah.
can do
Which is-
therapeutic work
yeah,
on fetuses.
For the benefit of.
Right,
For the
right.
benefit,
but-
right, you can do it, and there's no limit on the risk, so long as you minimized it as much as possible.
Right, right.
But it doesn't mean that it has to be minimal
But,
risk.
but I mean, I'm envisioning research where you're going to be discarding these embryos. I mean, you're not going to implant any of the research embryos. So you're talking about when you get past the
There's a
preclinical.
point.
Post-IND.
Oh, I mean,
There's a point at which you will, right?
I think there could be a point at which you can then start getting federal funding. But all the other stuff leading up to that, yeah.
You could start at the point at which the FDA has approved an IND, and we're now working to try to make pregnancies happen.
If you're starting to transfer embryos, I guess
I just want to make
so.
sure I really understand this.
Oh,
'cause
yeah.
it's
So
just like- it's weird.
I meant it only to apply for the in vitro stuff, the
Right.
preclinical leading up to justification for it.
But it's linked to killing another embryo.
Well,
That's the.
The mitochondria come from another oocyte. If they come from
depends on
another
oocyte.
oocyte. But if the mitochondria come from an
Yeah,
right.
oocyte, then you haven't destroyed. But
Right.
that also
It depends on
is
the
relevant
technique.
to which technique you use, and Pat King's point about avoiding destruction of embryo.
No,
right?
but still, your efficiency is only likely to be, what, 10% or something? So you're going to be destroying
Well,
90 of, you're not going to be transferring 90% of the embryos.
But the prohibition is on creating an embryo for research purposes. It's not a prohibition on, unfortunately and unintentionally, losing
that's
lots of embryos
IVF.
along the way.
Otherwise, that's IVF. All right.
Are you a law professor?
No, you're watching. Yes, you're watching sausage being made, which is never pretty. All right. One last question, and then we need to close up. Oh, it's two questions. Sorry. This is from Jonathan Kimmelman, so I'm going to read this from an email that he sent. This is to the FDA. At what level of granularity does FDA review toxicology and proof of principle studies? Are you making approval or no-hold decisions based on summary reports of preclinical data that's aggregated, full research reports, actual experimental data submitted by the sponsors, or how? And then I'll read the second after that.
Okay, so for the IND review or any regulatory review, we will require the complete study reports, not just only the summary. So the level of the details for the IND review is different from like a grant review. So we need more details for the complete study report, not only summary, but also individual animal data for each parameter that is evaluated in the study.
Okay. So the second question is about requirements, if any, about replicability of preclinical studies in support of new INDs. So in particular, in high-risk realms, does the FDA ever request strict replication of proof of principle?
The short answer is yes, because for phase 1 clinical trials, safety is for safety. It's safety clinical trial, but it's also a benefit risk evaluation. So if a specific product has, let's say, more toxicity or and the study population is not life-threatening disease, then we do need to say some proof of concept studies in order to balance the risk benefit ratio. So the proof of concept studies is also part of the this risk benefit ratio element. So it depends on the bar of the toxicity profile.
That's helpful. All right. I think, unfortunately, we have to close this session so that we can get to our folks who are coming in from overseas. Thank you all. And we need to shift gears at this point. So this is now going to be all by web.